Good morning, everyone. Welcome back after this lovely afternoon yesterday and evening. I hope you all had fun and could socialize a little bit and discuss a bit more about the topics that we raised yesterday. So yesterday we started the day talking about the communities and give you some idea about IBOL and EBP and their European nodes. And we discussed a lot about how when biodiversity genomics meets policy and practitioners and their needs. So a lot of what we're going to discuss today in this panel was actually initiated yesterday, which is great because it's going to give us some fuel. But just to get started and give you a little bit of introduction to this first session, which is supporting mainstream deployment of biodiversity genomics in Europe, we're going to have two short presentations, starting with Kristian Meissner and then following with Amber Scholz. And Kristian is going to be talking about the implementation and standardization of molecular monitoring methods. Amber will follow, talking about capacity building in biodiversity and genomics, observations from the policy-science interface. So we're going to get started with Kristian. Thank you very much, and have fun.
Thank you, Camila. Okay, this, okay, let's see. Oh, it works, great. So, what is a standard? So, I want to make clear that when I talk about standards, I'm not talking about some guidelines or de facto SOPs, but I'm talking about a standard that is a de jure standard, something that has been taken up into legislation or is very close to being taken up. As standards that are annexed in the Water Framework Directive. And for me, standardization is not equal to harmonization or intercalibration per se. For me, a standard is a document that has been established by consensus, approved by a standardizing body that provides rules, guidelines, or for characteristics or activities or their results aimed at the promotion of optimum community benefits. And the important thing here is I'm not talking about research. So all of you blue sky researchers that are resenting standards, this is not for you. I'm talking about the use of standards for defining minimum criteria so that these methods can be used in regulatory settings for monitoring, say for biodiversity targets within the EU environmental acquis or UN conventions or whatever, but not research. So ideally, the process would go like this. We have a new technology that is really promising, doing something, and then we do some standards easily, and then we take care about accessibility, data sovereignty, open access things, and somehow, automatically, they are taken up into policies. Well, ideally, it would be like that. But the truth is, it's much more complicated. The reality is that we have three different streams that are independent of each other and not often touching one another. One is the problem streams. In our case, when we talk about molecular methods, it is the lack of money, it is the lack of taxonomists that are getting older, and also just the lack of producing enough data to address the triple environment crisis. And then we have the policy stream, which is the solution stream, where the methods are developed. This is mainly for, say, the researchers that are doing their bidding there and developing these methods and are really keen on doing that. And separate from that is the politics stream, which is a totally different beast that works on defining legislation, agreements, and so on. And this is these are not naturally not touching one another. So then we have the Horizon program, and we have projects like EDN Aqua Plan, like this one, where we are now, and DN Aqua Image, that try to combine the the policy stream with the problem stream. Often they have also elements that touch on the politics stream, but they're not really doing a lot to align these. What really is doing that is, for the problem stream and the policy stream, is standards. In a word, a standard is the combination of the problem stream with the policy stream. And that is because it is a consensus agreement that has been reached through experts, and they agreed on putting this forward. So there you have the combination of these two streams already. Then the important bit is to convey a message to the politics stream that now these two other streams have touched. They are there, so to speak. And this is done by people that are termed by Wallin and Hultema, hopefully I'm pronouncing that right, the policy entrepreneurs. These are experts that are interested in doing work around getting policy into action. And what they're doing is they're doing a sort of a transcoding in the middle. So they're trying to align these streams and merge them to one, so that the outcome then is the policy that they're gunning for. In this case, eDNA being approved for official use and legislative monitoring or whatever. And that is a consensus agreement. Then we have another player here, Ecostat, somewhere in between. Actually, it's a bit misplaced, but it's on the verge of the window of opportunity where these streams can meet, and then in between the politics stream and the policy stream. So somewhere in there sits Ecostat for the Water Framework Directive. So when we look at standardization and how well we are doing with respect to standards for eDNA methods, and I'm using eDNA now very loosely as molecular methods, really, that can be used for the identification of taxa in a setting for the Water Framework Directive or Marine Strategy Directive. There's been a lot of activity in the last 2 years since I have established this group, ISO-TC Working Group 13. This group has produced already 5 outputs. So 5 of these outputs are already in the standardization pipeline. They're not official documents yet, but they're very close to getting there. So they're in the making, so to speak. And this is unprecedented. This is very, very fast, and it's not normal. And this is done through a process called that is ISTEF. I will not talk about ISTEF here for lack of time, but we have a platform that is very conducive in putting these forward. Number 6 and number 9, now in the blue, these are very close to getting to the pipeline. So in 2 years, we have almost produced 7, if I count correctly, standards, and we're producing in the next 2 years, I'm guessing somewhere in north of 5 new ones. So now, back to on the right, this diagram, we had technologies, we have standards, now to the accessibility and data sovereignty and open access thing. Well, first, let me say, before we get there, Ecostat has taken this on. So this group that is thinking and contemplating about the methods uptake for Water Framework Directive has established an eDNA task force, and Steinar is here in the room. I saw him earlier there, and there are other members here. Florian Lese is coming today, myself and others are members in this task force. And this task force is holding a key stakeholder workshop on November 21st, and I'll send a link around after this meeting on my LinkedIn account, so you can take part in this. But it's really important that this is also taking and also getting to the politics stream. And about the politics stream, so there is something that the United Nations Environmental Programme has been preparing for a couple of years, which is really on the accessibility, data sovereignty, and open access of environmental data. They put forth a data strategy, which is now being in the second draft and will be discussed at UNEA 7 in December, which is the Environmental Assembly of the UN in Nairobi. And there, although that this working group 13 that we established 2 years ago is just very, very young, is already mentioned as a good example on how to do this in the space of eDNA. So we are piercing into the politics stream now with our messages, and it's good that we do, but this is something that we need to do more. And then, in closing, I have to really stress this. This is a team effort. It is not for people with a strong ego. This is for people that are team players. So if you want to get these methods into policy, you need to be a team player. No ego will help you. As the African proverb says, if you want to go fast, go alone. But if you want to go far, and in this case, to the policy outcome, go together. And that was basically what I wanted to tell you. Thank you.
Thank you very much, Kristian. We're going to hold questions for later during the panel. So now I'm going to invite Amber Scholz to give the next introductory talk.
Perfect. All right. Thanks, everyone. I would say I'm a little bit, oh, I gotta go forward. All right, that's me. There we go. So I'm a little out of field, I would say. I spend most of my work time working on a couple of United Nations instruments, the one that's most well known to this group, the Convention on Biological Diversity, but also the new High Seas Treaty and the World Health Organization and the Plant Treaty. But in that context, I keep bumping up against biodiversity and capacity building. And so Camila and the organizers had said, you know, give us a whirlwind tour of what capacity building and biodiversity genomics means. And so I'm putting together things that I don't have any depth in, but that I thought maybe could be useful to trigger your imagination. The starting point where I wanted to bring you in on is, why do countries at the political level, and this builds off of your talk, fund capacity building? Where is that sort of motivation in the first place? And actually, you can see this in the KMGBF itself. So at the very end of COP15, the Chinese presidency basically said the only way that we're going to get the global biodiversity framework over the hill to conclusion is to have a take it or leave it deal. Very Chinese approach to policymaking, but basically they said these 5 documents is the package. If you don't want it, you get it's all or none. And you can see in the structure of that the things that most would say that the northerners or the environmentalists sort of want are the first 2 things. That's the goals and the targets of the biodiversity framework. Then it's the monitoring frameworks, the quantitative approach to get there. And then the things that many would say the global south sort of wanted was capacity building, resource mobilization, that's the money. And then this thing that I work a lot on called digital sequence information that we're not going to talk much about today. And that tells us if we want to think about where communities like those that are brought together under BGE will need to position themselves to be part of the larger movement of where and how and why capacity building for these UN instruments is done, then you need to be on the whole package in order to get there. Yesterday, Gilles had mentioned the TSCs. So within that number 3 there, within that capacity building plan, what policymakers started to do in 2022 was to say the way that we get to capacity building in the new biodiversity strategic plan is to build these regional and sub-regional technical support centers in which we specifically focus on the achievement of the goals and the targets of the GBF. Meaning, if you want to be part of capacity building, you're only going to be in the in crowd if you're part of the headline indicators and the goals of that larger GBF. So what was said yesterday by Kristian and my question sort of towards the end of the day is if you're not on the headline side, if you're not on the mandatory side, you kind of don't count. And for now, eDNA, metabarcoding, and the broader use of biodiversity genomics is on the nice to have part of the KMGBF, and it's not part of the mandatory stuff. And I think that's a gap that this community could think about as we head towards 2030. The other policy angle, and these are the two slides on digital sequence information, this is a paper that came out by a colleague and I earlier this year, which is where and how that fifth thing on the KMGBF, the digital sequence information bit, landed ultimately. And so what we love about the new digital sequence information and how benefits from genetic data are going to be shared is that we, through a lot of discussion from the scientific community, were able to have open access to digital sequence information and to explain how and why that contributes to non-monetary benefit sharing as well as to capacity building. But in that box up there on the upper right, that's all the accidental capacity building. That's the stuff that we're doing anyway, that nobody's sort of paying us to do or that we're doing on the margins of existing grants. The real question, the policy question, was whether or not any money from the new Cali Fund, once money gets in there, that should come from the private sector, whether any of that money would also flow towards the purposes of capacity building. And if you take a look at how the text during COP16 last October, when we operationalized all of that stuff on the previous slide, you can see how this evolved. So when we came into the meeting after the open-ended working group meeting on DSI, it said only that money from this new global fund should go to IPLCs. Then about halfway into the meeting, we got very excited because some of our ideas to the DSI Scientific Network had landed into the text, in particular this call for 10% of that new global fund being dedicated and set aside for capacity building. And there, in other language, you could see that it would also be focused on the use and the utility of digital sequence information, genomics, et cetera. However, you can see that once they went into the closed-door negotiations, that was deleted. That yellow text was deleted, and what we ended up with in the final document is a proportion, meaning you can do it or not. Why? Because the political compromise that countries had to make is if the money can either go to me as a government or it can go to capacity building, I'd rather have it go to me. They couldn't say that in front of stakeholders, that's why they did it in closed negotiations. But ultimately, if we want to have more money head towards this for the broader overarching biodiversity goals of the planet, then we have to make sure that we can compel even or communicate to countries that it is in their own national interest. And I'm still trying to figure out how to do that. And so now I wanted to give you a little bit of a whirlwind tour. I'm going to go fast. I'm happy to share these slides of just examples of what capacity building in the broader sense of biodiversity have looked like over the years and how I think that they have evolved with the idea that should the BGE and the broader community want to use these as models, this is just a collection of ideas and examples. So the first one comes from Japan, which is an international joint research program called SATREPS, and they have brought together many different sort of life sciences themes together, including under environment and energy and bioresources, where various types of biodiversity programs have been funded. And you can see these always have the component of being at least bilateral and most often multilateral. And here again, you'll see basically Japan will commit as a country at an international conference, you know, X number of tens or hundreds of millions, and then it takes that money from its ODA, from its overseas development aid, it funnels it through several different agencies, and then funnels it out to these bilateral capacity building programs. A little bit different is the Biodiversa one, and I think this is on the edge of whether or not this is truly a capacity building program, although I think it sees itself at that. It was mentioned several times yesterday, and this is probably the #1 place that you can get large international biodiversity consortiums funded, although I would also argue here genomics is still only one thing of which they do, and they're still very, very close to the existing sort of policy frameworks that don't necessarily include genomics in their focus. This is a bit tricky. Yeah, exactly. And then here's just some of the areas to give you a feeling that, you see projects to policy, internationalization, private sector, and then you can kind of fit some of the things we've been talking about over the last 24 hours into these first two, but a general scan makes it look like that's not widespread. Oh, I'm running out of time, Camila. Here, the Americans, for a number of years, for 15 years, actually funded some of the very first large-scale international biodiversity programs, again here through 3 different federal agencies, even though the US is not a party to the Convention on Biological Diversity. And in fact, this is a little bit of their blood money for not ratifying, is to say, but hey, we're still one of the major international funders of biodiversity, and it was through programs like the ICGB, BGE. But these were, again, really before omics was taking over. Then a few examples where I think you can see capacity building and omics technology, but not necessarily where there's a clear biodiversity emphasis. Another one funded again by the NIH, but in partnership with the Wellcome Trust, as well as, again, this sort of co-creation model with the African Academy of Sciences, was called H3 Africa. This has much more of a health focus on malaria and other neglected tropical diseases, but very exciting, the amount of omics that was in this one. The Periodic Table of Food Initiative, funded by the Rockefeller Foundation and a number of other partners, focused on metabolomics and food, looking at small molecules, extremely exciting, the size of data that they're building up. The Cabana program, funded by the UK Overseas Development Aid agencies, but then through them, focusing on biodiversity as well as communicable diseases, and very focused on Latin America. And here you can see some of the partners as well as colleagues from EMBL EBI. This project ended in 2021, which was a crying shame because it was amazing, but has carried on in a smaller context to continue on training. And then here, kudos and shout out, because IBAL was basically able to, if you will, repackage itself and truly land on the international capacity building for the CBD under the auspices of the Global Taxonomy Initiative. I would say for the EBP and the ERGA communities, you haven't gotten this far, so I would challenge you to sort of get into there, because IBAL has gotten that far, which is really sets you up more for that policy interface and for the larger pots of international money. And then finally, gosh, I'm sorry, this is a bit, there we go. Just some ideas on what we've discussed within the DSI Scientific Network, topics that we have heard from our international members around the world, what they need when they do omics. And that really focused on training at the very, very, very top, to produce DSI, to use DSI here. DSI stands for Digital Sequence Information, for infrastructure capacities and also organizational capacities. Next on our list was the idea of biodata centers of excellence. You could maybe also call that infrastructure, but infrastructure that's not focused on global, but on regional needs, regional priorities, noting that the vast majority of omics-related databases are based in the north. And that means that the vast majority of those operators of that infrastructure are also based in the north. And this call to diversify that, which was also mentioned yesterday, that the INSTC is already listening to those calls and diversifying its international portfolio. But yet there are still very significant gaps. And then finally, these ideas of matchmaking platform. Here's an example from Costa Rica that has been very popular and very successful in Central America. That's my concluding slide, Camila. Thanks for the patience. What I really wanted to do was maybe to say the questions that I can't ask because I'm on the panel, which is the broader question: does the policy mandate that we have at the international and at the regional level to do biodiversity genomics, to need biodiversity genomics, does that exist yet? Because I would hypothesize that you need that mandate in order for large-scale biodiversity genomics-focused capacity building to be called into existence. This relationship between biodiversity genomics and bioeconomy, for example, the CBD's second objective after biodiversity conservation is sustainable development. This is a sustainable development treaty, and supporting the bioeconomy, this intersection between the two is, I think, we can't forget it. And this discussion we had yesterday about whether or not these technologies are really ready to move from the research agenda into the broader mainstreaming. Yeah, thanks, Camila.
Thank you so much, Amber. And I already took a picture of your questions to add to our panel. So it's my pleasure now to invite again Kristian and Amber, and also Tiina, Laamanen, and Andy Nisbet to join us at this panel. So the idea here is to make it somewhat dynamic. So I'm going to just throw a topic and let's see how it develops. And if the audience has anything to add immediately, just raise your hand. But I will get to the point later where I will ask if you have questions or new topics for us to discuss here. And let's start with standardization. And I would like to know the opportunities and challenges that you in the panel see for bringing these different molecular methods, barcoding, metabarcoding, SNP-based approaches into mainstream monitoring programs, taking the standardization topic into consideration. Who would like to start?
Well, I mean, I can start, but I'll start. So, of course, I see a great potential. Otherwise, I would not engage in the whole standardization business. And I think what I try to convey in my talk is the necessity for us to convey the message to the politics stream that these methods work to fulfill the criteria that are already set forth in legislation or in the requirements of programs. And they do that. So these methods can be retrofitted to fulfill these requirements, but they can do so much more. But I would go with a shoe in and say we can retrofit, and once we have convinced them that this is doable, then you can open the closet behind you and say, well, we can do all of that as well, and then show the whole potential. So show that it works for what you're doing right now, and then explore the potential gradually as you become established. Do not try to push the rope, is my message.
What did you want? Tiina, maybe?
Yes, I fully agree. And one thing is that because we all know that these methods, they are developing so fast, but once we are willing to implement this into our environmental monitoring, so standardization is the key. So we have to have that kind of consensus, that what is the minimum level what we need to do. Researchers, you can always do more and study more, and standards, they can also revise once we have more information. But at this point, we really need to implement these methods, and for that, we really need those standards.
Andy?
Yeah. So I think I mentioned yesterday I work for Natural England. That's a government agency in England looking at natural environment, and I lead several large national monitoring programs. So for me, thinking about how we can start to adopt these technologies, standardization, I guess, is critical because we have to have confidence in the data that we are generating from these, and in particular, it has to be reproducible or replicable, however we choose to frame the standards, so that if we are commissioning a survey this year and we get a set of results from it, and we are commissioning a repeat survey in 5 years' time, we know that the differences, or we can be confident the differences we're seeing are a result of a real change in the environment, not because people have used different technologies, different lab processes, or different bioinformatic pipelines. So standardization is kind of absolutely critical for end users like us to have confidence in this. But I think there's also a question of how we standardize. So Natural England and DEFRA, the government department in England, was kind of introduced one of the kind of first statutory standards around the testing for great crested newts. And that's been very successful, but in some ways it has kind of fossilized or ossified that process because it dictated the kind of the laboratory process and everything else. So there is definitely and I get a lot of people saying to me, we should review this, we should change this. Everything's moved, everything's changed, and everything's developed so quickly. this is outdated, we should do it differently. And then I talk to my kind of delivery colleagues and they say, no, this works, it's fine. We don't want to muck around with it. So there's definitely a kind of a question. We do need to standardize, but we do need to think about how we standardize so that we don't fossilize things.
Yeah, I think I would like to provoke a little bit because one thing, and I'll go back to Kristina's talk yesterday about genetic diversity and how this was added to the Montreal Global Biodiversity Framework as a headline indicator, as the effective population size should be higher than 500. And there are many ways to estimate that. Definitely, there is no standardization behind that, but this got into the headlines. And my question is, how can we make it all happen at the same time? Because, I mean, we have no time. So we need to convince that these techniques, because they're so powerful, they should be considered, and the information coming from them should be considered. But at the same time, we need to make standardization happen, and as Kristian said, as a community, right, really connected and involving as many different communities as possible. How can we make this happen at the same time so that we can convince the politicians, but also enter with the information in the correct way, so that it's really informative and not even misleading? One thing that worries me, that everyone will be doing something in a different way, and at the end, we're just deviating from what we should be doing because our results are going to be wrong or different.
I mean, I there we go. Yeah. I can't answer anything about standardization. I'm too far out of field at that point. But the terrible truth, and you had this on your slide, I love these three arrows coming together, is that the hardest part for science is that translation, to know who are the people that are making the decisions about the thing that really, really matters, and to be able to filter out all the other stuff that you don't have time for. Because you have to do regular jobs, and those are good that you're doing regular jobs, but in order to find that little flip to make that thing happen that actually has huge implications for an entire field, you have to be in the right place at the right time and have the situational awareness to where that was happening. And I mean, just to give you on this specific thing for genetic diversity, because I have a little bit of a guilty conscience. So what happened is in 2022, we had a monitoring framework, but it wasn't done because parties realized there were a couple of places in there, including on genetic diversity, including on Target 13, access and benefit sharing, that they didn't actually know how to quantify it. They didn't know what they were supposed to do. So what do whenever they don't know? They send it to a working group. They send it to an expert group. And as I mentioned yesterday, Belma and I were on that. I was nominated by Germany because I'm an ABS expert and I have a methodology to collect global data on non-monetary benefit sharing, which we can talk about another time. Belma, I don't know, why were you there? Because you really know something else about not that, right? Or it was genetic diversity, but we were in the room in this small group of 5. And I remember sort of trying to sort of say, yeah, but this won't actually fit all of them. We actually need to have a couple of different options, like what about this, what about this? But then there were people that were farther afield and sort of said, no, but I think it's supposed to land here. That's what like the group consensus is. And you kind of get steamrolled from a group of, I don't know, 30 people. And this kind of groupthink, I mean, I think, and I don't want to say, but in this particular context, like looking back now, like if one of you had been like, no, actually, this is the language that you should use to be able to give yourselves the flexibility to have molecular methods captured in the monitoring framework, and I would have been happy to champion that for you, maybe Bowman, right? But I didn't, but it's very hard to know who to ask because it was midnight in Montreal, and who were you supposed to ask? So this is the hard part about that science policy interface.
Absolutely right on. I think I couldn't agree more. It is really about knowing the people that make the decisions, but it's also talking to them. I mean, it doesn't help that you are crying out in your own field that it's a crying shame that these methods are not taken on. You need to engage with these people that make the decisions in real life. I mean, that is what you need to do, and you need to convey your message. And here again, my point is the standards give you a tool that you can say, you know what, all of us here, the whole world has agreed upon this. This is something that we agreed upon. This is consensus. You can use it if you want to, and I can tell you why you should. And then you make your point. And once you make your point, it becomes much more easy, but you need to time it. Like the window of opportunity on the slide, it needs to be on the timing. I mean, these people are flooded with information, also with lobbyists and so on. So you need to establish trust, you need to be at the right time, and you need to have your homework done, i.e. you need to have standards or something that is more than just a nice presentation. And that is really helpful. It is really difficult. It's not easy, especially not easy for scientists that are not used to doing this sort of work. And this is why this particular paper that I cited, Pauline and Huitema, they call these people translators or transcoders. So people, experts that really engage with the politics stream and do that work and try to pitch. But it is something that a little step out of your comfort zone as a regular scientist.
So now we understand that there is a long way, or we need to connect to policy, so as scientists, and we need to make this conversation go. But when talking about standards and developing them, and the example that you gave on water standards, water monitoring standards, what are the challenges, and what do you think we have now a route that all the different fields can follow? So you made for water, can we follow similar paths for soil, for insects, for marine monitoring? What are the challenges that we still need to overcome in order to get together and develop these standards? And that goes to any of you, of course.
Okay, sorry, do you want to go? I mean, I touched on one of them earlier about the kind of the risk of fossilizing processes. So the challenge is being able to standardize without doing that. I think there's a kind of question of how to, or which ones need formal standards, because not everything needs that. You know, there's a lot, you know, when I think about this, the kind of work we do, there's a range of work from regulatory stuff, which may end up in court, which has certain evidence standards we require from it, all the way down to kind of local projects thinking about nature recovery and kind of nature restoration. Not everything needs a kind of formal standards. Some things just need kind of best practice and guidance. So kind of an understanding of what we need, what level of standardization we need for what purpose and what areas as well would be valuable.
I can share one good example how you can get people on board. So Kristian mentioned ISTEF, so it's International eDNA Standardization Task Force, and it's open to everyone. And it has been really good example on how to get scientists and all the experts on the board. So we need that kind of mechanisms that everyone can feel that they are involved and they can provide the expertise. And of course, one thing is that technological readiness level, that how to proceed. And for example, in water eDNA, we are already quite close to full-scale implementation. And I really see that, for example, airborne eDNA, we will follow within some years.
Kristian?
Yes, I mean, the great thing about these methods is that there are no national traditions, because they are a real problem. Once you have established national traditions, you're in a swamp, because everyone is, oh well, it's more like a warfare along trenches, because everyone is keeping their ground and not wanting to budge a millimeter on their things, because they would have to change. But there is nothing to change here. There is only something to be preemptively defined with these methods. And that is the great benefit. But the big danger that I see now is, you ask, what can we do to do it with other fields? What we shouldn't be doing is that we should be establishing 6 different ISTEF groups, or similar groups, for different fields that work in silos, not talk to each other, do their own stuff, and try to influence the policymakers. because then we are just, again, all over the place and not reaching them at all. We should now try to converge into several mechanisms and build them and solidify them such that they can be used for different purposes. ISTEF as this model, I mean, it need not be ISTEF in the name, but it doesn't matter really, but the model can be replicated and can be enlarged to cover terrestrial, aerial, whatnot methods. Doesn't matter really. The big thing is inclusivity. Amber mentioned it. I mean, the global south is outside. They have all the biodiversity. What we are talking really about is just trying to see that our technologies are somehow solidified and then we can push them. But it is about global biodiversity. So the South needs to be involved in these discussions. And platforms like ISTEF provide that possibility. You can engage with these people that tell you, you know, doesn't work in our country because we lack minus 80 degrees freezers. And the whole continent of Africa was told there are just 5. I mean, that gives you a perspective for some sort of methods are out of question there. You need to think differently and perhaps also lower your bar on where your minimum criteria are for these sorts of standards then. I absolutely agree with Andy. Not everything needs to be standardized, but for the important, the regulatory stuff, those things need to be. And that is what I'm trying to do. So not everything, really do not think like that.
Very nice. I think this starts to make an interesting bridge to capacity building. So Tiina mentioned this readiness in technology, which is something that, I mean, in order to, if we have standards and they demand the use of certain technology, the different countries have to be ready to use and to apply these technologies. And this goes from the capacity development, so the technology should be there, should be implemented properly, and also the people, the experts, should be trained and should know what they're doing. And this goes, I mean, we're talking the European context where there is a lot of differences even across Europe. This is something in talking about genomes. So we see a lot of difference because the genomic at the genome level is more expensive than what usually you can do with eDNA or metabarcoding. Like you can make approaches that get it very like cheap as an approach. For genomics, or genomes, we are on the way for that. But what does happen is that some countries do not have the same access even to the technology as other countries do. And I think this is, I can say that for some countries in Europe, it actually mirrors a bit what happens in other parts of the world and even in the global south. And which I find quite interesting because this is a very interesting connection that we can make. So when we, as an European consortium or community approach the European countries that have limitations, we are also developing methods that can be used in other parts of the globe, because we have to live with, and adapt to what is possible. Like we cannot impose technologies that are too expensive. We cannot change certain realities, but we can adapt our methods and making sure that these standards will still work even if they don't have access to the same types of methods. So then I would like to bring this as so we are talking about capacity development, so in the sense of also infrastructure, and on the other side, capacity building in the sense of training. So what do you think, once standards are there, or even maybe for the development of standards this is necessary, what do you think are the challenges in terms of developing standards and having the need for capacity building? So how to follow up with both together? Kristian, would you like to start?
Well, on the capacity building, just to think, I mean, Pedro would be much better to talk about that. He's done that. And I don't think you should limit yourself thinking about Europe. From the get-go, you should involve African nations or something in other nations from the global south in your project, at least as a mirror for what you're doing to get the feedback. So be more co-creative and not so Eurocentric in your approach, but rather look further. I mean, there is a world outside Europe. It is hard to believe sometimes, but there is a world outside of Europe, and it's much bigger than Europe, too.
Have I told you I'm Brazilian? So yeah. We definitely. When I say this, it's actually from experience because I have a consortium with Brazil, and we are making a connection between Latin America and ERGA through this consortium, slowly. And I can really see the similarities, which is something that I find quite interesting because we can approach the problems in Europe and can connect directly with what is happening in Latin America in that case, and I'm sure in Africa and Southeast Asia, for example, as well. Yeah. So, but yes, definitely, we should not just think about Europe. We should think globally. But a lot of when we talk here about this consortium, it is already a big challenge, right? We handle 38 countries. And with a lot of differences, so it is a big challenge already.
If you have 38, what do another 100 matter? I mean,
oh, sorry.
If you already have 38, what does another 100 matter?
Well, yeah. Okay, yeah, Antti?
There's somebody up there as well. But I'll say something first. So I've got quite a different perspective in that mine is quite parochial. It is England focused, it is one country. And I think my thoughts around capacity building, if I think of our staff, we have 2,500 staff working on nature conservation. Most of them have no training in this. They don't have any depth of knowledge in this. We have a handful of people working nationally who do have some expertise. If we want these methods and techniques to be used routinely, to be business as usual within NGOs and agencies like ours, then we have to develop the skills of our staff so that routinely they are able to let contracts, look at reports, understand data, and particularly interpret what they mean in terms of what decisions they should be making, what actions they should be recommending. So for me, a lot of this capacity building is not about us. It's about the people who don't understand this technology yet, but are going to have to use it if we are mainstreaming it.
Good point. I think, yes, someone from the audience, could you? This one?
Thank you. Birgit Gemeinholzer from Germany. I'd like to support you, and I think there shouldn't be that big divide, especially the genomics and genetics tools should link to the morphology and all the knowledge that is already there. I think we shouldn't divide that is the new tools and it doesn't link to the old methods. It should be a transition. It should be a soft transition. Of course, we want to develop the new tools, the new standards, but they need to be interoperable. And the way where they meet are the databases. And especially GBIF, the global international database, it's being used by researchers, by citizen science, by policymakers. So I don't think the gene banks, like you said in your talk, are the right places where people look, because they're only scientists look at the gene banks. But at the level where you have the connection to real world life, to practitioners in all the different levels, is GBIF, and I think we really should promote that.
Absolutely. And I think there is already quite a tight connection with taxonomy, for sure from the barcode point of view. And I think, and this is something that we, at least as BGE and the eyeball ERGA, have been working on establishing connections to other areas that are looking to biodiversity. And I mean, these databases are being developed, and there's many projects that are interconnected. But you are completely right. I mean, doesn't make sense, and we should not isolate genomics. It is just that, in many cases, genomics is something new. It comes with big data. It comes with bioinformatics, and so people become afraid of using it. And so there is a barrier to overcome in terms of technology and these first steps of capacity building, but we should not isolate it at all. I agree with that. Would you guys like to comment on that as well?
Well, I can say that, yes, I fully agree. And for example, what we are doing in Finland currently, we are developing that kind of like a national level solution, but straight away linked to international databases, because we see that we really need to build it that way, that whoever needs that information, it doesn't matter, is it like from molecular analysis or traditional analysis, there should be that kind of place where everyone who needs that information, it's easily available and it's really fair. So that's something that we are currently working really hard, like a national level and also international level. For example, yesterday I mentioned a Horizon EU project called eDNA Aquaplan. Many of colleagues are also attending, and maybe, for example, Katarina, you can tell more about what we are doing now, but the aim is to really make that kind of like a digital landscape that we see that what is out there and how it could look in the future, that everyone can access to that data.
Amber?
Yeah, I've been sitting here. So in my previous life before science policy, I was a microbiologist with full genome scale perspectives. And one thing that I've noticed about these recent discussions is that this automatic, assumption, I think, that many are making, that the mainstreaming will at least start with, and my question would be, will it be limited to the barcoding world? Will the mainstreaming that we're talking about here and the standardization, is that going to stay for the foreseeable future, focused more on the barcoding types of approaches? And what are the opportunities, or are there any good stories to tell about barcoding approaches or eDNA, and then you need the full genome because X, Y, Z? Are there any examples of standardization? Are there any examples of the need to mainstreaming? I mean, I think there might have been one or two yesterday, but I'm wondering whether and how that narrative can be developed for that interplay, because one of the really amazing things about BGE is pulling those two communities together in novel ways. But yet in these panel discussions, and recently, I kind of feel this gravity towards the barcoding side of things. So I don't know if does that community get to go first, and then the full genome community comes next, or can they come of age together, and if so, what would the narrative be?
Excellent question.
Can I come back
would you
to
like to?
So I think the barcoding community is inevitably going first already. So for me, for my world and the kind of what I'm in, I can see how we use this, how we use barcoding in monitoring and kind of widespread monitoring, either in existing monitoring programs or in new ones that we might develop. That's really quite easy for me to see, and it is relatively easy for me to explain the benefits and to sell that. I'm not a molecular scientist, so I guess I kind of struggle slightly with the genomics world. Yesterday afternoon helped quite a lot. There were some really good examples of why this makes a difference, how we could use it practically for nature. But I think we need more of that, and I need to kind of understand, if I'm going to sell, Natural England is not paying for my time to come here because I find it fascinating science. They're paying because they think, okay, we'll be able to use this to make better decisions, to get better data about nature. So I think I need a more convincing case. I need to be able to make a more convincing case, or a fuller case, for the genomics and why organizations like us should be interested in investing in it.
Kristian?
So we had a workshop on genetic diversity just a week ago, I think, in last Friday. International guest Gernot Segelbacher, you may know him, IUCN and whatnot, in many places, very involved in genomic diversity, coined it rather well to the crowd, and the crowd was not pleased, saying that genetic diversity is a niche of a niche field at the moment. So, and that is the plain truth. And I'm sorry to say, the same week I had a session at the UN General Assembly science summit where Jan Martinsson from the JRC was saying that decision makers don't care how the data is derived. They're just interested that it does work. So there is the gap. You think you need to tweak everything down to the last millimeter. That's not true. Decision makers couldn't care less. They just want what you showed, Amber, in these documents to be fulfilled, and that's good. It's our duty to make sure that the foundation that we build on is sound, but we need not be over diligent with this task. It needs to work, it needs to be sturdy, but we should not overdo it, is my first message. And I'm not disparaging the community of genetic diversity at all. I think it's a very important thing, and we need to be including it, but we need to also see what is workable. And I think, Annie, you're quite right. It is much easier to sell the barcoding first, and then as we have the shoe in the door, we sort of sneak in the genetic diversity later, because that comes later in a way. So we can just show this works great for this, but did you know it could also work for that? And then open up the closet and say, we can do all of this and more once we are there, but we need to get there.
Oh, wow. So many people. Yes. I'll let, one second, I'll just make a comment, and then you go, you guys, Pete and Rob and Brad, so everybody, that's a great point in time actually to throw to the audience, so that's wonderful. From one side, I see this as well, eDNA so much easier to enter, in the different spheres, to sell it as something that is useful, that is fast, and so on, than when we talk about genomic information. But that's, I always say this is the difference between community ecology and then population biology, when you really think about what is happening interspecifically at the boundaries of populations, on the management of a species. This is when, one of the sides of when you really need genomics and you really need to enter on the genomic diversity side of things. And we have to make it simple. And I think this is something that the Coalition for Conservation Genetics, started doing, bringing a simple message that not necessarily is bringing genomics with it. And I think it's our role and duty, to bring genomics in the simplest way that we can, in order for this to be easy to understand and very unacceptable and easy to use as well. So yes, we are coming, let's say, behind, but hopefully we're going to come in parallel. And this is also why BGE is important, because if barcode can break through, in BGE it's bringing genomes with it. Yeah, so now I'm going to throw to the audience, and we already have a sort of a queue there. You can start.
Yes, thank you. Nice discussion. I really like, to see like what the people from the policy side and scientific side are considering. In a way, I represent a little bit the bioeconomy side of this. And what we are struggling with at this moment is that we are operating in the now and are already applying these methods a lot. And we are struggling a lot with the standardization challenges now, and also the way this feeds back to legal obligations that we have to our customers. So I was wondering, do you guys already think about how we can, on the medium term or short term, already work with something so that we don't get presented with something that is very different from what we are now working with to make it work for now? And particularly, I'm would be interested in things that we, for instance, to make it a bit more concrete, we are really one of the big challenges that we are looking at is standardizing and normalizing data sets through space and time, and also normalizing for things like PCR inhibition, because these affect a lot the comparability of data. So yeah, I would like to see your response to this.
Okay, who would like to start?
I'm sorry, again, me. Yes, well, first of all, you're welcome to contact me on these issues. point out what the difficulties, where the difficulties lie. I will be sure to take it up with the experts group that are now working on standards and ask them whether it makes sense in their view to have that standard. Also, you're welcome to propose it yourself. I mean, if you're interested in going forward, this is an open community. ISTEF is open to everyone, literally, that has a stake in this. So this is what the important thing is, that we get the community, industries alike. The IOGP, the International Oil and Gas Producers, have now guidelines on the use of eDNA for specific monitoring stuff. It is a shame, in my view, that they did their own protocols. We need to get these sorts of players involved in the standardization business such that they are adhering to the same standards, so that we send a single message and not multiple ones, because that will confuse. It's like arguing parents. If you have arguing parents, the kids don't do what they should be doing it. So.
Okay. I'm not sure I can follow that. So I guess I had two responses in the short term. One is to kind of get involved in collaborative partnerships, either nationally or regionally. So in the UK, we have the UK DNA Working Group, which brings together researchers, businesses, and public sector end users and NGOs. And that's a forum for building capacity and having just those kinds of conversations. And through that working group, Natural England has done a piece of work which is set, I'm now reluctant to use the word standards when it's not appropriate, but has set kind of metadata guidelines for when we're contracting. Because we recognize one of the kind of challenges is this drive for openness, drive for standardization versus private companies' desire to protect their intellectual property. And how do we kind of reconcile those? And so one of the things we've done in consultation with businesses is say, okay, if we're letting contracts or if you're submitting data from third parties to Natural England for regulatory purposes, these are the metadata we expect as a minimum to allow us to scrutinize that and to have assess the level of confidence we can have. So those sorts of things, I think, are helping us in the short term while we work for standardization.
Yes, I fully agree, and we really need events like this and that kind of discussions that we are on the same page, that what are those bottlenecks at the moment. And by the way, there will be eDNA Aquaplan workshop discussing, for example, data issues. So can you, Katarina, say the dates?
Yeah, we also want to find out from the people at the workshop, not just what they think of the ideas in principle, but will they do it? And if they think it's a good idea but won't do it, then why? So that we can then not just submit another report to the EEC about from a Horizon project that will end up in a black hole somewhere, but a report that says, to make this happen, this is what you should do. Next week, Wednesday, Thursday, online from 2 to 6 CEST.
Thank you. I will now give the word to Magda.
Yeah, thank you very much. So I'm a forest geneticist, moving slightly towards connection with policy world, and coming from a Mediterranean region where we don't have this lux to have a community working only on barcoding and another community working only on genomics. Myself, I started with genomics, studying genetic diversity in many species, and moving later, joining Paul Hebert IBOL organization. And I use them as a tool when it's necessary. If I want to know who is here, what is in our ecosystem, I use barcoding. And when I want to dig more into adaptation of the plant, what to plant, when, taking into account the climate change, etc., the responses is in genomics. So I feel it's a little bit artificial problem opposing or competing between these two. Especially when we reach out to policymakers, we should tell them, we do monitoring, we spot something, and the response is coming after we have a community working on it. And just a small comment, I learned lately that policymakers don't like to be influenced. They like to be informed and guided. And we don't want to make a consensus before, otherwise we will be really late in coming up. So the work of an institution like mine, European Forest Institute, connecting knowledge and action, is to not make the consensus, but to make a kind of synthesis. This is the status of the science today, and these are the repercussions. If you go this way, this is what you face. If you go this way. So just make also a little bit from this new thing I learned about informing and guiding and being ready to support them, to take their decision, because they have a lot of other elements. More than science, they have society, election, agenda, etc. Thank you.
Excellent point. Brad, would you like to follow up?
Hi. Some of you know in this room, some of you don't, that one of the projects I run is a large project called the California Conservation Genomics Project. And this is relevant to exactly what you were just saying, but also to the sort of eDNA versus genomic side of things. So we skipped the eDNA part entirely and went straight to landscape genomics, 250 species. We've generated 22,000 whole genomes to characterize genomic variation across the state of California. Now, it's just one state in the US, but it's a big state. It's got the lowest point and the highest point in the continent, in the lower 48. It's a very, very, very diverse place. And I think part of the discussion I've been hearing is that, well, you know, policymakers aren't ready to absorb that, or it's hard to pitch it in a meaningful way. And what I did was I took a very simple concept, which is to say we have California is a biodiversity hotspot, one of the global original 25 hotspots on the planet. We have tremendous biodiversity. We can't manage for all of the potential extinctions that are going on by using within species diversity as a proxy for the ability of species to evolve their way out of problems rather than us having to take care of them. We can identify patches of landscape that contain the most overall among species genetic variation. Those are the places we need to protect, and with any amount of luck, they'll be able to evolve their way through the problems we're throwing at them. And everybody loved it. I mean, members of the state legislature, very powerful politicians, they said, this we can understand, this makes sense, here's some money, go do it, and we have. So I don't think it's an impossibility, and I really don't. So anyhow, that's my, now it is just California, and California is a unique place in many ways, especially right now in our country. But it's also an exemplar. I mean, it is a very complicated state politically. It has, you know, people from all sides of the political spectrum, people who are environmentalists and people who are very much not environmentalists. It's a very challenging political place within the U.S. And so I think at some level it is an exemplar of a broader community.
I really am happy for you that you got your project and that it works for you. I'm just saying that in the EU, given the political context that we're in, given the environmental acquis that we're in, where it is very hard to get to that. So because we have to have consensus across the member states, which are, it's like you had to do the same project on the level of the US, and I think you would be less lucky then because you could not make it. What we need, though, is in Europe, we need these sorts of examples of being able to demonstrate that we can do it in one of the member states for others to follow. So having a sort of a pilot of sorts, and then follow that. That is a really, and that might be much more easy to get the one member state's ministry to approve your funding for something like that. It is a totally different game on the European scene.
Can I mean, Brad, I think that's super useful. It could, again, I'm always kind of, or I feel of it as the outsider. And I can't quite say what your narrative was in a 1-minute elevator pitch. So I think what you said, let me see if I can try spontaneously, extemporaneously, is for certain biodiversity problems where we already know these are the things that are definitely about to die and we clearly have them on our radar. Genomics is going to be a better tool, or full scale, because we got to know what's inside the genome and how bad it is. In other situations, like many of our other mainstream examples, where we want to have much bigger picture but lower resolution, then we want these barcoding tools, and they're complementary. But no, you're shaking your head, Rob's nodding, I love that. So because what my question was earlier was, like, what is the narrative? Like what is the if this then this, and if this then this? Because if you want to speak as a biodiversity genomics community to diverse policymakers, they need to, in a nutshell, understand, okay, like this is a toolkit, and these are the tools I grab to for this kind of a thing, and for this kind of a thing I grab to these, ideally matched up to the targets and goals of the framework. But anyway, that's the what I'm missing as an outsider.
Sorry, I tried to say it, Amber. The narrative is that we cannot manage our way species by species out of the biodiversity crisis. What we need to do is discover if there are regions where within species variation is high enough that we predict species can evolve their way out of, let's say, global warming. And if those pockets of habitat exist, then those are the ones we need to protect for biodiversity. And then we can just leave them alone. And they will evolve their way through, and they will provide the solution for those species to avoid extinction in the face of climate change. And the eDNA side of it is, as many people have said, at least in my view, that's about finding out where things are and where things aren't. And monitoring, which is completely different. It's one thing to say a species is here, but its genomic variation has been so depleted that it's entering into an extinction vortex, and it's not a healthy population. So it's about genetic health and resiliency is the big word.
So it's about triage. I mean,
It's a,
here, and then you guys can triage it.
well, or you can just protect it and then just leave it alone.
Okay.
Just-
Sorry, Magda.
Yeah, just make me think at European scale, we have U4Gen, European Forest Genetic Resources, where you have these genetic conservation units that are exactly what you are defining. That are some places, some forests where we don't have yet characterized genetically, but we know that there's potential genetic variation enough to ensure that they will be here for the future. And this is where every country in Europe, I mean, a lot of them are in this network, and this is one good example of this.
So, yeah.
And
So can I just come back? Yeah. So it really kind of links back to what I was saying earlier. I was struggling to see the kind of case for it, and that helps. So, but as well as the elevator pitch, policymakers are going to want the 50-page business case as well. So So I'd be really interested to see what that said, because in the end they're going to know. I keep saying it, but they're going to want to know, how is this going to let me make better decisions? How is it going to be economically beneficial? How is it going to be strategically worthwhile? So yeah, I'd be really interested to see how you pitch that.
Could you identify yourself? Sorry, it was just
sorry,
you.
yes.
Whoever is going to talk, please tell your name.
I'm David Eckers from Waardenburg Ecology, ecological consultancy company. I find it very interesting that you are talking about like taking measurement effectively to kind of like see to what extent the system is resilient and is capable of doing some equal evolutionary dynamics to adapt to future conditions. And we as a company are also interested in this, but one of the hurdles that there are in practice in actually making this work in the real world is developing tools that will make this affordable to measure and scalable. So we are looking at like maybe using qPCR on indicative SNPs to do a more affordable way to get there. But like I would really like to invite also the scientists here to try to think also on the application side of many of these very promising and important things that we need to measure to assess sort of the adaptability of the ecosystem. But how can we actually scale this and afford this? Maybe California is very rich, but like many countries in Europe where the budgets are small or regional. So would like to hear something about the response.
We have an interesting little corner right here.
And
so
sorry, Brad, and then you can pass your microphone directly
behind,
backwards.
that'd be
behind
great.
you.
And I should identify myself again, Brad Schaefer. I'm at UCLA in California and run the California Conservation Genomics Project. And I mean, the people talk a lot about the price, and you know the price of sequencing has come down so low at this point. I mean, so low that you don't want to say it's not an issue, but a lot of organisms you can resequence a genome for $20, $25, $30. I mean, it's really quite low. Okay, if you're doing hundreds of thousands of organisms, that's a different problem. But I think that's one where I would agree. You don't necessarily want to resequence everything, or maybe you do, but you know, you certainly don't need to. One of the things we've done in the CCGP is I hired a team of amazing postdocs who built, you know, really, really clean, clear workflows for analyzing data. We published a paper a couple of years ago with a workflow called Alligator, which takes the workflow directly from the bioinformatics pipeline, plugs it through 15 or 20 different very standard landscape genomic tools to be able to sort of, you know, push out the other end the kinds of maps and you know, integrated landscape maps of genetic diversity and stuff that people we think would standardly want to use. So that's all there. It exists. It's all freeware. So we've done some of that that I think makes it fairly approachable. And you know, I mean, we're going to be wrapping it up in the next 6 months. We've got essentially all the empirical work done, most of the analysis done. Now we'll see if science wants to publish the papers, but it is doable.
I actually wanted to comment on that because I want to even refer to Leif's presentation yesterday. And what is the best way to use genomic information, genome diversity information? In the ideal world, you should do what I already mentioned yesterday, the genome enablement of a species. Ideally, you're gonna recreate, or let's say establish a basis for the genomic diversity of that species, all the different populations, or at least as representative as possible, because that's the best way for you to find the SNPs that are really informative for whatever question you might have. Once you have this baseline of information, you can redirect to very specific questions and then develop assays that can be super simple. And like SNP chips, or even some multiplex crazy number of markers that you can do at once. Yeah, so there are many different approaches. And I think this is something that we will focus on the next step, let's say, of BGE, if we get funded. And that's where we're going to connect to the other EBP. So the California project is also an EBP project, and we are going to connect to the different projects to see what everyone is doing so that we don't all recreate the same thing. And so this is really important that you take the first step on focusing on establishing this baseline of genomic information in order to then make genomics informative and feasible, cheap enough to be able to use it. Rob.
Yeah, thank you. So Amber, I was nodding while Brad was shaking his head because you were heading in this direction of the kind of precision tool, the precision, like the analogy with precision medicine, where we're using reference genomes and full whole genome resequencing to get a very detailed view, but on a very specific, very small slice of biodiversity. And I think what Brad is doing is bringing that into this kind of mainstreaming world where we can actually look at not a whole ecosystem, but representatives from that ecosystem. And that's what we'd like to see more of, right? So we were not disagreeing per se. And we need more pilots. We need more examples like that. So in Europe, we actually do have one pilot at least. So in Switzerland, if we count, which was a fantastic pilot project with just 5 species, but sampling across the entire country to bring, like Camila was mentioning here, these baselines, right? We need more baseline examples so we can see when a particular species actually is in danger. Because we have an idea, but we need actually examples of perfectly healthy species to contrast with less than perfectly healthy species to understand where the thresholds lie, where the dangers are, and where it's actually now the moment to have some sort of intervention. And there I would circle back and say that the genomics is then enabling us to measure the impact or the effectiveness of such interventions in a way that other technologies simply cannot give us that kind of level of resolution. So yeah, we were agreeing to disagree.
I can move to Pete, Rob, directly.
Yeah, I mean, just to follow up on this point, I think with the whole genome data, we've got the Global Biodiversity Framework Target 4 on genetic diversity, in which it's measured using census counts as a proxy for effective population size, as a proxy for genetic diversity. So we've got this kind of nested set of proxies. We have the ability to measure genetic diversity at scale now, and a policy framework requirement for it, and early studies that actually go out and measure it using genetic data will be really valuable. And I think that's one kind of key route in. While we do the much wider conceptual stuff, we've never had this data before. Like the human genome, like all the advances for human medicine, there will be a whole bunch of things that we're yet to find out about applications, and we should also be very comfortable that that's a journey we're on, but get these early, easy wins.
You guys are free to answer. I just want to say we have 5 minutes, and I would like to maybe shift a little bit towards the future, thinking about how is biodiversity monitoring going to be in 5 to 10 years? Will genomic approaches be fully integrated? So you can answer or comment already the questions and comments from the audience, but please already shifting to the future. Yeah? And then I would like also a comment maybe from each of you so that we can wrap up. Tiina?
Oh, well, yesterday I showed our vision, what we made once we were making our national eDNA roadmap, and we really see that these methods, they are almost there, and within 5 years we will get there. We will combine molecular-based information, we will combine remote sensing, modeling, so, and it's something that we really, as a community, we have to do it, because as we all know, we so urgently need that information, so there ain't any other choices. So we have to do it, and I'm optimistic that we are getting there. Of course, different methods, they are following a little bit different timeline, but in the end, I'm really hopeful and optimistic.
Andy?
Will we get there in 5? Well, Maybe. I think it depends by what we mean by getting there. You know, inevitably, I don't think everything is going to be mainstreamed by that point. You know, I think the eDNA and barcoding approach will be much more mainstream. I suspect the genomics approach is still going to be not as mainstream until we can make those arguments, and also think about, as you kind of mentioned, you know, there are policy windows where opportunities open up and then they close again, and we've got to be prepared for those. But some of them may not open up for several years, but we should be ready. I guess the other thing I would say, in order to be able to do that, I'm just amazed no one said reference libraries yet in this panel discussion. But you know, until we can get, you know, for us, until we can use stuff with confidence, until we have those well-curated, reasonably complete barcode reference libraries, it won't mainstream. We can't mainstream it. So that's probably my number one priority still. I know we keep banging on about it, but until we get it done, I'll keep banging on about it.
Thank you for mentioning the importance of reference libraries. We from BGE agree very much with that. Amber, would you like to?
Yeah, I guess I have two perhaps closing thoughts connected to that question. My first was a reaction to what you've been told by policymakers, that they don't want to be lobbied. Nobody wants to be lobbied, but welcome to democracy. Yeah, I mean, I think that this is like an age-old question, the interface between science and policy, and I do think that what we can do very well and effectively is basically to say, if you do this, then this happens. If you do this, then this happens. Or if you choose this path, then you lose out on this. And if you choose this path, then you lose out on this. So to sort of provide that objective framework. But also then, once a relationship is built that bridges that sort of science policy divide, then absolutely you should be giving your unfiltered advice and recommendations. And it's been my personal experience that you usually sort of start off in that more objective, neutral way, and then once that relationship develops, then absolutely it is the job, I think, to give unfiltered, like gut-checked recommendations. And that brings me to sort of this future question: will genomics land it in 5 years? And my read of it is that the global biodiversity policy cycle is a 10-year cycle. I mean, the Convention on Biological Diversity meets every 2 years, but they have, at the 2020, that was pandemic, that's why it ended up being 2022, but at 2030, they're gonna kind of look at it all again, you know? And it takes a long time to get from that narrative that Brad and Rob were kind of going back and forth on, and that elevator pitch and the ideas here, to be able to land successfully in an international policy document. And so my personal recommendation for, if this community wants to be part of the mainstreaming, is you need to start now to get that pitch down clear. Like, biodiversity genomics can do this, it can do this, it can do this. And to your point, it can do it cheaper, better, more accurately, with less labor cost, AI-ready, digital transformation. I mean, you know, pardon, I guess it's being web streamed, you bullshit bingo the entire one-pager, right? And, but that takes, you have to field test that for a year or two before you can tell what's landing in order to get it to land. And remember, you know, parties spend a year developing their position to go into the negotiations in 2030. So really, you gotta be ready by 28, 29. So I definitely see the potential. I have learned a huge amount, and I'm very thankful to everybody here, but I also think you're wiggly on a couple of spots in that narrative, and I think you can strengthen that, and I think you can do it together. So I'm optimistic, but you have work to do.
Thanks. Kristian, and then we close.
I mean, what they said, and then some. But basically, Amber, I think you're dead on. It is about conveying the message. Someone said in a management context, if you say something to your employees, by the 7th time, about 20% will remember what you said. So that is, you need to hammer in the message over and over and over again, and then hopefully it sinks with some of the crowd. Don't expect one pitch to land it successfully. I mean, there's Brad who's done it, but the others will probably not succeed. The policy cycle, very important. It is long, and it needs a lot of preparation, and you need to get your shoe in there first. I'm a little less optimistic than Tiina with respect to full-blown European implementation by 2030. What I will say is that by 2030, we will have a decent amount of countries that are piloting these methods and showing that they work, and I'm hopeful that by 2035, the EU is on board with meter barcoding as one possibility for getting tax information into the Water Framework Directive context. With respect to other directives, I'm hesitant because it needs some more convincing. There is more legwork to do there.
Well, I would like to thank our panelists. Very enriching discussion. So everyone, we have a break now at the VIP room, and we should be back at 11 in the same room, please.